The overall goal of this procedure is to perform continuous live imaging and single-cell tracking of multiple neural cell populations, followed by post-imaging immunocytochemistry. This method allows the analysis of any cellular events that take place within the tracked time period.
This method can help to answer key questions in the neuroscience field, such as cell behavior and lineage progression of multiple neural populations, as well as the identification of their modulators. The main advantage of live imaging and single-cell tracking in real time is that it allows to monitor variations in cell biology that may pass unnoticed in approaches based on endpoint analysis.
This demonstration will guide researchers through main steps that can be easily adapted to any culture condition and acquisition software.
My name is Rosa Gómez, and I'll be demonstrating the plating and post-imaging immunocytochemistry steps.
My name is David Agustín-Durán, and I will be demonstrating the live imaging part of the procedure.
My name is Lucía Paniagua-Herranz, and I will be demonstrating the single-cell tracking section of the procedure.
To begin this procedure, seed the cells directly on poly-D-lysine-coated 24-well plates, with 1 milliliter of culture medium per well. Incubate the plates at 37 degrees Celsius and 8% carbon dioxide for adult neural stem cells, or at 37 degrees Celsius and 5% carbon dioxide for the astrocytes or cell line for 2 hours prior to live imaging. Turn on the microscope, camera, hardware, and incubation systems. Set the temperature and air pressure to 37 degrees Celsius and 8% carbon dioxide for adult neural stem cells, or to 37 degrees Celsius and 5% carbon.